Good evening, children. This is my third video on cell cycle and cell division. In my previous video, I had started with the meiosis division, and as I discussed earlier, meiosis is to be described under two headings, meiosis 1 and meiosis 2. And in my previous explanation, I had already explained about meiosis 1, consisting of prophase 1, the longest, consisting of 5 substages, metaphase 1, anaphase 1 and telophase 1. Today's topic is meiosis 2. So, this diagram at page number 10.15 I have already described, children. So, after the prophase, this all is prophase, children, A, B, C, D, E and F. That is leptotene, zygotene, pachytene. This is continued of pachytene, diplotene and dikinesis. What we broadly saw, just to summarize in the gist, is in the leptotene, the thread-like structures were there with increased nuclear volume, right? Then chromosomes appeared to be longitudinally single, but DNA had already duplicated. Zygotene meant pairing, because zygosis means pairing, and here the pairing of homologous chromosomes was seen through synapses, and this formed bivalence. Alright, then each paired synapsed homologous chromosome is called a bivalent. Then comes the next is pachytene stage. Here the bivalent divides by a longitudinal furrow. Each component of the bivalent divides by a longitudinal furrow and forms a tetrad. Alright, so each tetrad consists of 4 chromatids. You can see here 2 black and 2 white. And then takes place the most important process of crossing over. So this crossing over, I had told you, the portion of each homologous chromosome breaks and joins into each other by getting exchanged. So the breaking is by an enzyme called endonuclease, and the joining, called as annealing, is by an enzyme called ligase. A very important structure formed is X-shaped structure called chiasma, right? Then after this stage comes this next one, that is diplotene. And in diplotene, what you can see is another important process of disjunction and terminalization. Right? So disjunction meant that the synaptic forces, they were getting lapsed, all right? And finally, the chromosomes, they became increasingly shorter. By coiling, they moved further, right? And the chiasma moves away. So this is what you saw here as terminalization, that this point of contact is now gradually becoming to the terminal ends. So that was our previous time's division. Moving on to the last step, dikinesis. This was the stage, that is the end of the prophase, and during this the chromosomes continue to contract, and the chromosomes, they are the thickest at this stage. Alright, then due to terminalization, their bivalence appeared rounded, and the centriole and centrosphere divided. They moved to the opposite poles, and the nuclear membrane disappeared. You can see here the nuclear membrane had disappeared. Then came metaphase 1, the same equatorial plate formation you could see. Anaphase, the splitting of the chromosomes. And finally, in telophase, the nucleolus is recovered along with the nuclear membrane. Also, the chromosomes, now they again start becoming thread-like. So what the division started with in telophase 1, you can see the chromosomes uncoil and form the chromatin net. That all we did last time, children. It was a long discussion on meiosis 1. Today's discussion is about the second part of meiosis, what we call as meiosis 2. Meiosis 2 is comparatively smaller to discuss than meiosis 1. Here it is simply prophase 2, metaphase 2, anaphase 2 and telophase 2. See, broadly speaking, the main purpose of meiosis is done in the first division only. What is the main purpose of division, children? That the diploid chromosome number that started with 2n has already become haploid. That is, each chromosome material here has already got the haploid number n. So what is the need for chromosome to divide? What is the need for the cell to undergo meiosis 2? And the answer is, in this case, children, we agree that the chromosomes, they have reduced to the half, but they have double the amount of DNA still. That is, each chromosome, yes, it has come to half the number, but still it has double the amount of DNA, which now will actually become into true haploid nuclei at the end of meiosis 2. So let us begin, children. This is very simple to understand. Prophase 2 begins with three major changes. Number one, chromosomes reappear. Previous, I just now told you, they had again uncoiled in telophase in meiosis 1 to form the chromatin thread-like structure. So here the first change is the chromosomes again become visible as thick, short and stout structures. Nuclear membrane disappears and spindle formation begins. Let us see this. Three things. In prophase, the chromosomes are clearly visible as their true structure, that is thick, short and stout structures. Number two, the nuclear membrane starts disappearing. Alright. And then the third is the spindle formation begins here. So you can see this is the starting of spindle that has already begun from prophase 2. Metaphase, what happens? Three major changes. Spindle formation is completed. Chromosomes oriented at the equator, forming the metaphase plate, right? And the arms of chromatids are directed towards the poles of spindle. So let us see here. Spindle formation started here, but it has completed over here. Second, the chromosomes aligned at the equator parts in a plate-like structure. And the third is that the arms, they are directed towards the poles. Alright. Then third stage comes anaphase 2. Like previous mitosis only, the chromosome divides and the chromatids separate and they start getting dragged to the opposite poles, right? So let us see this. Just like any mitotic division, from the metaphase, now there is some pull on the centromere which divides and the chromatids start moving like the same inverted V-shaped, right? And to the opposite poles. Coming next to the stage, what is telophase? The chromosomes of each group uncoil and develop a nuclear membrane that are organized to a nucleus, and this is followed by cytokinesis. Finally, reaching the telophase, children, the further division is same. If it is an animal cell, division is by a furrow formation. If it is a plant cell, then it is by cell plate formation. But you can see that the nuclear membrane is recovered, the nucleolus also comes back, and the cells are now rightly divided into two daughter cells. This daughter cell complete division is done after the karyokinesis. This is how it is followed by the cytokinesis. So the meiosis 1 is very simple. Once again, let us revise. Here the nuclear membrane disappeared, spindle formation started in prophase 2. Metaphase 2, the spindle formation completes. Then metaphase plate, the chromosomes align at the equatorial plate, arms directed to the opposite poles. Anaphase, the splitting of chromosomes takes place to the opposite poles. And telophase, the nuclear membrane is recovered along with the nucleolus. The division completes of the nucleolus of the nucleus. This is followed by the division of cytoplasm, and finally the end product is 4 daughter cells formed. So this is the complete discussion about meiosis 2. What is the importance? The importance of meiosis 2, I have already told you that this actually makes truly haploid nuclei, because chromosomes, they had already reduced to half in meiosis 1, but they were having double the amount of DNA. So the real meaning of haploid nucleus that is formed is here, when each daughter cell is truly haploid with the chromosome number that is half. The significance of meiosis is, number one, formation of gametes, the sex cells. Then 2. Maintenance of chromosome number and stability of species. See, it is important that during the sexual reproduction, 50% of parents' chromosomes and 50% of 50% of fathers and 50% of mothers' chromosomes, they are normally aligned to the right number in the progeny. So this number of chromosome stable number is achieved by the meiosis division. Third is introduction of variation, that is different amount of combinations that are possible in genetic variations. They are because of meiosis. And finally, the interrelationship between organisms, that is the details are essentially similar in all sexually reproducing organisms, and this shows the basic similarity and relationship among all of them. So that's all about meiosis, children. Today we started with meiosis 2. There are no notes to be sent because the discussion we held from our diagrams in the book. Please read it twice or thrice. If there is any query, you're free to ask, and please drop in your queries anytime. Thank you.